Welcome to our tutorial today. And in this tutorial session, this is about histopathological imaging, and I'm calling it a histopathology and imaging because it's not solely imaging. So we will go through some of the basics of histology. I did show you a video of some essentials. That was just the basic, and we will cover that a little bit. We will not do a video, but we will go through some of the major processes. And then we will try to look at some of the basic mechanisms by which imaging is done by basic processes. And I may not list, I have listed them, but will discuss maybe some of them, and we'll look at some images. And also I'll tell you how when we screen when we look at the sections, how it is that we count the cells. How do we decide which field to look at? So let's look at all of that together. So a little bit background about pathology, about histopathology in itself. There are about four facets to pathology in itself. One is why would we call this histopathology? So it's all under the branch of simply pathology. One is the etiology that helps to define the cause. Pathogenesis is what leads to that whole disease, and that could be molecular in nature, and it could be also risk factors, etc. So all of those consists of pathogenesis. So it's the mechanism of the whole development. Morphology, on the other hand, is just structural changes. So looking at morphology by whichever method. So it could be the whole organ, it could be just the tissue, or it could be the individual cells. And then finally putting it all together is giving it a clinical significance, and that is relating it to activity, relating it to function. So we need to interpret these microscopic images as should I give Joydeep the co-host? Can somebody
No,
tell me?
I think
It's
you
fine.
should go yeah, with Shantanu, madam.
No, he's already his co-host. Okay, so
Okay.
we're fine. Okay. So where was I? Yes, interpreting the microscopical changes is really a not a very easy process. It's a very specialized process. He's still not admitted in. Okay, I'll let Jaideep in right now. So it's a very specialized process, and the thing is that there's the only word to it. It's very subjective. We're not saying that it is error prone, but it is subjective. And for that reason, histopathology always has to be done by independently by the histopathologist, different histopathologist for the same slide. And those are protocols that are set in so that there is no subjectivity, and then if there is any bias, then that is caught. So we look for tissue patterns. We already know what tissues look like, and so we look for these various patterns that might be slightly different than what is normal. So histopathology is um, is is slightly superior to just single cell cytological samples because there is that relationship with all the tissues, the all the individual tissues, they need to be together. So it's like saying, if I do cell culture and I have individual cells in a petri dish, then looking at them is kind of not the same as looking at the whole tissue. And the whole tissue is because they all have some kind of relation to each other. So especially in cancer, this is much more true, especially in cancer, because you need to look at the stroma, which is, as you remember, the stroma is all the the surrounding cells and the surrounding um, tissues that surround the tumor, and so it is kind of like we do independent cytology samples separately, where it is very convenient to take out to scrape out the samples and to look at just some abnormal cells. But when it comes to a tumor, when it comes to a cancer that is spreading, then it is really important to have the histopathology, which is looking at the full tissue with various tissues. It is looking at the relationship of all the cells rather than single cytological samples, as was done in a pap smear. So, and doing a staining a tissue is a fairly much more complicated process, and so that required several more intricacies in order to be able to look at the tissue as such, or the even the organ as such, and having it stained, and then uh, tying it towards tying it to staging, which we talked about, tying it to staging and to gridding of the cancer. So everybody has to be in sync, in synchronization when it comes to this. So there are a bunch of steps, and Uh, among the steps is to treat the tissues, but at the end of it, it is basically examined under a light microscope. What's happening? It's examined under a light microscope. There are much more fancier microscopes with higher magnification, but once it has been stained, a light microscope serves the purpose. It can be done, the histopathology can be done, the tissue can be processed by various methods, but a light microscope is one of the best tools. And so there are even better tools that we will also look at. And some of them, require creating an image. So we already said this before, histopathology, the whole report is based on a interpretation. This interpretation skill is from the expertise of the histopathologist. And so it has some subjective value to it. So um, why do we do this? Why is it that for cancer tissues, we don't necessarily just rely on molecular markers and more on blood tests? Why is it always that cells are always taken and examined, especially in cancer? Well, number one, I want you to remember that the primary tumor is slightly different from the secondary tumor. The primary tumor is a bunch of cells that have formed a tumor, but when they travel and metastasize, they go at a secondary site and form a tumor there. So it's kind of important in a cancer to look at even a secondary site and to analyze where the tumor originally came from. And so, in order to do that, really, microscopic examination is probably one of the best ways to do that, and with additional tests. And so, why not? Because it's number one, it's kind of like much less demanding to begin with. However, we do have, yes, we do have it gives us the ability to look at the architecture of the whole cells, which is very important. That is why I talked about the primary and the secondary tumors. And to do histology, it is like this. We have a section of the biopsy. We can have just take another very close by section and simply add different stains and look at it differently. So we can the the flexibility in being able to cut additional sections once the biopsy has been obtained, once the tissue is at hand. So multiple multiple uses. Okay, somebody else is waiting. The co-host is not letting them in, I think. So so what are the disadvantages? Well, number one, it is a bit time consuming to create those sections. It's a whole different process, a whole different team, a whole different expertise to make these sections. And sections is, I'll show it to you. Sections are simply the tissue in a piece of wax that then is stored and then can be sliced and then stored. Um, so, but sometimes it is also a little bit difficult to separate out just by looking at it, looking by histopathology, just by microscopy and histopathology, some various kinds of cancers. And so, multiple ways of confirming are always important. Okay, I'll let this other person in. All the techniques in the pathology were major big growth pathology. Growth pathology means you have the full organ, looking at the whole organ as such, cutting out the, doing a surgery to really look at the full organ. And the rest is based on cells and tissues. And of course, there can be an autopsy. So it is mainly it is biochemical in nature. There's no doubt about that. It can involve, as you already saw, it can also involve antibodies. So antibodies are required when the cells they they um, they may look alike, and it is not possible to tell them physically apart unless they are stained with antibodies for a specific protein that the cell will have. And in that scenario, even if you have similarly a little bit similarly looking cells, having an antibody stains it slightly differently. It is much more expensive. Stains it slightly differently, and it can be identified as a unique cell compared to its neighbor. And most of the time for this, you would need not a normal microscope, but fluorescence microscopy. And finally, we can have the electron microscope. The electron microscope can be used for very, very niche areas where we want to really look at cellular level, subcellular level, organelle level. We can do that. And of course, molecular pathology, these days, we cannot do anything without looking at proteins, genes, markers, and it is getting faster and more inexpensive by the day. So it's really doable. So what is histotechnology? That's the whole process of processing the tissue. So that means really um, before the study. So that is all about making the slide onto making the slide eventually to make the slide and to be able to the technologies that is used for preparation of that of that slide. Okay, so histotechnologies can keep on changing. The technology, histotechnology can keep on advancing, but at the end of the day, we just call it histopathology together. And so here are some nice some pictures for you.
Recording
So
in
here are
progress.
some pictures of how these are done. And so this here, as you can see, is a cartridge. So the samples typically that they weigh that they are taken is that a from the biopsy there would be the edge of either if they're lesions, if they're oh, my this thing is white. Right, let me just okay, it works. So uh, edge of the lesions are taken because that's where you can see the normal cell versus the abnormal cell. So if there are abnormalities, and it could be a lesion or it could be a cyst or it could be a tumor. So some edges can be taken. The walls of cysts are typically taken. So now I'm talking about everything here. And you don't want complete normal areas. You want to be able to have, if it's a at the border, but you don't want a section that is totally normal. um, but you do want to include it in locations where you can compare. And so some sections will be completely normal, but other sections can have normal and associated abnormal areas with it. So together. Necrosis, you want to avoid a necrosis area. Necrosis is total cell death that is massive areas of cell death. There's no point in taking those, and those are not useful for the diagnosis. All right. Most of the time for us in research, for me in research, I we take the whole specimen, we take that whole liver or everything together. And why? Because it just fits into the cassette anyway. It would fit in the cassette once it's taken in. And so and there is a way, a methodology for which there is a sense of direction, and that direction has to be marked as to what side you are taking. So the types of biopsy, that means taking out the sample. Could be tissue fragments. That's called as a curettage. So that is done in a skin, and it can be done in the endometrial lining. A Another types of biopsy are for taken with a small gauge needle, and they are called as a pipal biopsy, where in the endometrial lining in women the sampling is done with that small needle, and it's really a very small amount. It's not a lot, but it's using a small gauge needle. The core biopsy is for using a large bore needle that produces a core of a tissue. So it's like you take out, it's like taking out an apple core, and you take out a piece, which is from the breast. So your idea is to have some intact tissue and not to have this the loose cells. In an endoscopy biopsy, for example, in the GI tract, um, they can remember I showed you the polyps yesterday, and I showed you how the polyps essentially they just get scraped out, and so if they get scraped out with the help of the camera, then you have the whole polyp to really to look at. So that is an endoscopic biopsy. A punch biopsy is taking a little piece, piece, and that's just the specific technological name is taken out from the skin. So it's all about the size that is required, and you're trying to get in some layers into the dermis. The incision where you're using a scalpel, okay, a scalpel to really go and incise into a lesion. You're really cutting into a lesion, and you're taking out stuff. And cone biopsy, also, although I never talked about it, it's in the cervix, and it is done by a um, special looking also scalpel, and it is in the cervix. In order to get deeper layers, a cone is taken out. So it's really like literally like a 3D cone, and that is the piece of the cervix. So a small piece like that from the cervix, it's called as it looks like a cone, and so that is completely removed and tissues are taken up further. So the biopsy itself has so many mechanisms of doing it. This is not to get loose cells, but to get a tissue. And so now those were the porous cassettes. This is the the cassette that I just showed you, and so these are kind of porous so that there are chemicals that can go in and out of it. And so these are your samples here, and all of this is going to be used. It's probably going to go into another cassette, and so that we have uh, multiple samples. Everything is just not going to be fit in one of them. They're going to be separated out. And the requirement of a certain requirement to be 5 millimeters thick. So they have to be a certain, if they're thicker than that, then this was going to get kind of crushed. So you don't want that to get crushed. And so therefore it needs to be a certain amount of thickness, which can be done um, by the expertise. Now, at the same time, it is possible that when it fits in here, whatever fits in here is comes out to be of that same uh, size, and it has to be kind of like centered so that everything there's going to be wax all around it. And it is in a formalin, which is a preserving agent for about, a certain number of hours. So it's typically left for a bunch of hours so that it gets fixed. And by fixing, it means that the tissue, the proteins kind of precipitate and everything just stays where it is. And time just kind everything just stays so that it doesn't decay any further. And it all the tissue architecture stays intact. And so if there are any, um, you know, um, temperature requirements, if there are any change of solution requirements, some of these are automated. And so after fixation, they have to replace all that formalin. And so I showed you the other day how you keep on changing it from 100, from a low amount of alcohol, and to you're trying to remove all that formalin. And so you keep on changing the concentration of the alcohol, so that all the formalin gets removed. And so these are the different chambers. These are the different chambers. So just some general stuff that I thought, you should just know here, that when this tissue comes in, okay, although this is not a major topic for us to talk about, but we have to remember that ethical concerns are always there, and risk for disease transmission are always there. So everything has to be done in a respectable, ethical, and risk-free method. So the methods of transport of the tissue is has to be appropriate. We're not going to go into the details for that, but I just thought I'd put that in. And everything must be tracked. Okay, nothing can leave the hospital, the clinic, the patient, whether the patient is waiting. I mean, especially for cancer, and for any section, there is no such thing as it just leaves proper recording. So it has to have if it has to be sent to the histology lab, then it has to be the right mode of transport. Either, it has to be shipped after it's fixed or before it's shipped in the cold. And so there's a lot of people who are arranged in this kind of arrangement. And so when I say risk assessment, risk is not for the human being because it has a diseased tissue, but is also to the tissue itself. The tissue itself should not be damaged in any way. So, ethical and risk concerns both guarantee how these tissues have to be sent. So, and all these details have to be there. There has to be, no matter what, there has to be clinical details and what fixative were used, and whatever the sample is and how much formalin it has. And so sometimes they can be fixed in one place and then the histology can be done at another place. The staining can be done at another place. And this is just to show you that, there has to be an ideal size for the block. Okay, so this one really came in right after that picture, but there has to be an ideal size for the block, and in order to be fixed, and that is all dependent on where it goes, how much solution there is, etc. Okay, there's just and the section has to be sliced and cut if it was too big, but with extreme caution so as not to damage kind of tissues that you want to look at. And sometimes they also get inked, and so there is a the place where that it gets cut, etc. So this is like a big piece. All right, this is a big piece, so they get the margins get inked, and that is a way for it to identify different locations. And that I just put this in because I wanted to show you that large organs have to be marked with for some kind of location. And so when these are stained, so we will, we are not going to go through the exact all process of staining because we kind of looked at it the other day. But when we look at slides, when we look at slides, we have to have everything has to have a the data has to be controlled. Everything has to be compared with a control. There has to be some kind of normalization. There has to be a counting at different places. So so the cells, and I'll show you soon, the cells, let's say the amount of cell death, if you were measuring cell death, necrosis, if you were measuring the necrosis, then it has to be, it has to be based on cellularity. So sometimes you're really counting the cells. So you are looking at regions and you are counting the cells. And so I'm talking about a cancer in this case. And so how many cells are completely dying with compared to the all the other tumor cells. So this is by cellularity. So we're counting kind of cells here. And then you are looking by area. So you can look at the field by area. You just don't look at just count cells where if a tissue there was half broken, some places have more cells, some places have less cells. So you also look at it by area. So percentage of non-neoplastic stroma. So is there surrounding tissue that is there, and what is the area? So again, when you do that, then you're using the tumor area, as a comparator. So the full tumor area, um, and you're looking at normal cells that are in the area. So you're just measuring it by area. Here was cells. You're measuring it by determining it by area. And some of the places they have a lot of mucin secretion, so there are many, even in pap smears. There's a lot of there's sometimes the way that the smear is done, that there's too much mucin, and that's not a good smear because in a pap smear you can get single cells, and so you can always go back and you can try to get separated cells. And so again, so there can be acellular mucin, so mucin that does not have any cell, and that goes by area, and then you have to use the total tumor area as a comparator. So now we go to the part we discuss um, where we discuss, let me just move my bar, some of the the basic methods and some of the extra uh, different kinds of methods of the imaging, and what the method is of the staining, and of course to get a different imaging. So the H&E stain, we talked about this before. That's that's really your gold standard, and it is that blue and that purple. The The purple is kind of like the background. Sorry, not the purple. The blue purple is the nucleus. It's bluish purple, so it is bluish purple, and that's your bluish purple. That's your nucleus, and then the pink, which is much more lighter, kind of a background pink. So the pink, like this pink. Oops, this pink, mm, this pink. These are all the cytoplasm, which you already know. And that is used. So it is used really for number one for cytology for individual cells, as we saw in a pap smear, or you can scrape your mouth. If you scrape your mouth, scrape, scrape your buccal cavity, or buccal cavity, and on the inside of the cheek, that will also give you single cells. If you scrape your gums, that will also give you single cells. So that's cytology for you. And for tumor grade, that means for sections. So for tumor grade, that means by looking at sections, you can determine the tumor grade. So it's one of the easier ones to look at to us for tumor staging, and looking at the borders at the margins of the normal versus the tumor tissue. And so we saw some of these before, so I didn't put, there's no need to go back to that again. And other ones that I also had said the other day was really mucin. You do need to sometimes stain for mucin, and to be able to see if certain cells make too much of it or not. And specific, sometimes special stains for liver fibrosis. They have other stains called as reticulin, and so they provide extra information. So, well, another method of staining, and this is like a traditional, when I say core analog method, these are not digital, they're analog, they're the old methods. That's not the right word to use, but compared to modern digital ones, those are considered as analog. So, frozen section microscopy. In frozen section microscopy, let's say that there's a surgery going on, and during that surgery, there is the requirement, the surgery, they removed the tumor, they opened up the person, and they removed, let's say it's an ovarian tumor, I'm just making it up. So they find the tumor, they remove as much as they can, and then they need to, immediately kind of like see the cells then and there to make some key decisions of what to do next. And so therefore these are doing used during surgery. Okay, so during. So a frozen section microscopy is kind of like a little faster. Um, it's a little it's a faster way to know right at that time, and in order to see where the margin of the tumor and the normal is. And so this is done during the surgery, for which kind of like means which kind of means that the surgery has to stop, the patient is still there, and they will go ahead and do the frozen section microscopy, so because it's a little bit faster, and in order to decide what to do next, and then come back to the surgery room and then complete what they completed. So come back means the technician will right away let know, but everything is kind of like halted at the time. So it is a slightly lower resolution than your normal, what I showed you just before, than your normal parafilm embedded ones. It is um the other ones, they have wax embedded. They in the cartridge. In the cartridge there's the tumor, and then there's a white wax all around it, and those are then treated very carefully along with that, that 50%, 200%, 100% alcohol, etc. So these are slightly lower resolution, but currently it's understood that a frozen section biopsy. Oh, sorry, didn't I put in the wrong text name here? The It is found to be almost like 90% similar to a H&E stain. So, so a lot of major decisions can be made in this way. So about 90% similarity. So even though it has a slightly lower resolution, it's very useful. And so this is an example of a frozen section and a paraffin embedded H&E stain, and you can just see that maybe the section that has is frozen and immediately determined. And I can't give you the full details of the process, but cryostat cryostat sections means that it's probably just put into something extremely cold and solidified, and then it's just cut into sections. Okay, and in cryostat, there has to be a special reagent, special substance that prevents the cells from changing their architecture. So that is why it's called as a cryostat. And so you can see that the details of the staining and the 40x magnification are very much the same, except for that it probably took some less cells. And yeah, that's it. Okay, so and so I'm going to just show you some images. Um, So, whole slide imaging and digital pathology uses the full um, the two oops about these sections that are paraffin embedded. Okay, so they are in a wax section in that cartridge, and these they convert everything into a standard size image. So these are called as slide scanners, and these scanners will take all of those, the full slide, and they will automatically just convert them and just save them. And so this way, in a situation where the expert doesn't come every day, the expert can look at it anywhere else. And so there are some FDA cleared uh, microscopic systems where the whole scanner, at one lot, all of these slides, they just get scanned, and that is for immediately, so kind of like primary diagnosis, for early diagnosis and primary diagnosis also. And those images, when they are all those slides, they are stained for these different proteins. Ki67 is a proliferation. It is a proliferation marker, and Ki67 just increases in those cells. And so in those image analysis, you can look for Ki67 for proliferation. So Ki67 index, it'll just show up in every cell which has that marker. Because it is multiplying, it will just show up. And so you can overlap these. And so just to give you some view of what these all kind of look like. Okay, there's a chat which I need to see. So these different components and constituents that are present are all the stroma, So everything, all that your tissues have. Some of them are ducts. Here, some of these are ducts. So some of these, so this is a duct, and this is a lining of the duct. Um, in the previous one, some of these, so we talked about these before in the previous ones. That's why I just kind of like left it. So Here, this is I don't have the background for then this one whether this was cancerous or not, but just to show that a full resection is good. So these are all the individuals. So these are like columnar epithelial cells. They're fairly big. You can see that they're columnar epithelial cells here. Um, also, columnar epithelial cells here, and where's in some situations these cells, if they don't have much of, they all of these have beautiful cytoplasm here. You see that you have a nucleus and you have beautiful cytoplasm and you have this duct. But in those that don't, some of them, this could be, I don't have a record of this one, but this looks like all the nuclei. There's very little cytoplasm and there's just nuclei, nuclei, nuclei. So when you have just nuclei, nuclei, and you have very little pink, then those are probably some of your tumor cells. Whereas whereas here they look fairly good. They are pink and purple, pink and purple. So I'm hoping to be able to give you some explanation. I'm also not a cytopathologist, but we all learn by basic some experience. Um, we do scanning only in mice, the xenograph that I talked about. So we look at it in those xenograph mice, which I will show you a couple tomorrow. So in how they look like. And so these are other tissues. So, like I said, they have to be measured. They have to be counted in a tissue section, at least like 6 different points. So if I were a the histopathologist, I would look at six minimum of 6 different points. At least that's what we do for animals. So it would have to be, it would have to be like six different locations at minimum in order to get even an idea. So you look at them by percentage of tumor by area. So you can go by area, just what I did right now, and you look at and then you count the intensity. You count the intensities. Um, you count the intensity of the dye, or you look at the see, this is now a tumor, so you don't just have abnormal cell and you just don't have dysplasia. You have just full on growth. And so you go by area, and then you have to count the individual cells as well. So you can go by area in terms of intensity, and you can then go also by cellularity and take locations and count the cells, literally count the cells over total cells as well. So I mean in terms of nuclei. So that's the way it would be done. So in terms of nuclei. So then assessment using only areas involved by the tumor, which is, hold on, let me move this bar. So this is an example of um, prostate cancer, and they are looking at places that have taken up. Do you see how this is more pink? You see how this area is more pink, and this area is more purple blue. This is more, isn't that the right blue purple, whatever the word is? So this is more blue purple. So that means that there are just much more nuclei here, right? So if there's much more nuclei, and then there's the cytoplasm is not that much. So if there's, when they multiply fast or they multiply, they don't have also time for all that cytoplasm. So based on this, that's why they have taken sections from here, and then they took sections from here as well. All right, from the pink location. And here, if I look at this tissue, you have, it's just a very clean looking homogeneous kind of a tissue. It has purple nuclei, but it has a lot of cytoplasm also. So, and these are a lot of cells, so I can't tell you the direction. And of course, where there are ducts, where they are in single layer, that's, I understand that they have to be, those are columnar epithelium, and they have to be slightly bigger. So that's why those are slightly darker purple. But here they look they're very non-neoplastic. They look pretty much pretty good. Versus here where there's like on this side where there's just much more bluer. I can't tell here just by looking at it, but they do not seem to be a very homogeneous kind of a tissue, and there seems to be much, much more blue in locations and not so much pink surrounding it. And um, so all the assessments that are again done. Um, so this one here, this is using the full tissue in the section, and this is just using, assessments, total assessment, just using the um, only areas involved by the tumor. It's a little difficult to understand how those are different, but for us, I think as long as we understand that we have three major ways of looking at it, we're looking at it by mucin, and so there is in the whole tumor, if they're looking at it, that means they're going by different locations, in that tumor, not just this one slide, but they are going overall to all parts and to going over multiple slides multiple times, different areas. And so that's where they find that there is no mucin in totality. And they find that there is a stroma by area is about 15%. And then they find that there is, throughout the whole tumor, that there is bicellularity, that there is no necrosis. So they did not find any place that the cells were really dead. Necrosis is the not a good way to die, but when there's a lot of injury, inflammation, then they can die by necrosis. And so in this situation, they don't find that. So I hope I was able to explain this fairly. So these are a little bit harder now also for me to explain, not being the a major cytopathologist, but looking at only 30-day-old tumors xenographed. That's pretty much what I look at. So, and this requires heavy expertise. So, so here, what some of these we are used to looking at, but some of these are using fluorescence microscopy, so different kinds of microscopy. So in these normal and malignant, so this is normal and in malignant ovary, so this is your um, ovary images. All right, so this one is the basic a basic normal. So this is an image. This is not your H&E stain. Um, OCT, I forgot the full form of OCT. But it is an image
Optical
that,
science, OCTs, I think optical something.
yes, exactly, optical something, and I've forgotten what that is.
Tomography one is uh, OCT.
Maybe so on and so you have to look at the um you have to look at A and C. So A and C. Okay. So A and oh sorry, not A and C. Yeah, A and C is that type. And
Madam, I
so
interrupting for one question. Very valuable. I think this slide is very valuable, and because I am interrupting you for the one question only. Already I have given a chat also
oh, is that
that
that was your chat? Yeah, did I answer that one?
yes,
Yeah.
yes, yes. That is my question is cell plus cell, that is the combination of cell is called is the histopathological image. But other than cell, what are the different constituents or the component are in the histopathological image as a cell biologist? I think some of the blood vessels and what are the others?
Some of the blood cells, is that what you just said? What did you say? Some of the?
From these cell plus cell, totally a lot of cells or the combination of cell is called histopathological images. So other than the cells, what are the different constituents are or the component in the histopathological image which are that is seen in the microscope by the pathologist or the cell biologist.
So we are talking about tumors right now, and so in the tumors, everything will be more cellular unless that there is a lot of connective tissue, unless there is matrix, like like bone, unless there is matrix like that. So typically in a normal cell biology, in a in an anatomy and physiology course, you would be looking at ground tissue. You would be looking at the ECM, the extracellular matrix, in a cartilage. So in a cartilage, you'll have a couple of cells floating around and a lot of background matrix. And so that those are all your secretions by those chondrocytes and by those fibroblasts. And but in a tumor, you will have the macrophages. You will have, so far, all these slides that I showed even last couple of days, you don't see a lot of cystic fluid. All right. So you don't see those fluids. So you see mostly macrophages, stromal cells, fibroblasts. So the cells that I talked about yesterday and the other day in tumor microenvironment, that is what you will see. And for the most part, you will see they will be very tight. So you will not see your collagen floating around everywhere. So what you will have is that you will have you'll have here, you will have patches. So you'll have patches of immense growth and other cells. If this was a bone tissue, but this is not a bone tissue, so that's why it's not here. So ideally, like I said, cartilage, you will have matrix, you will have your extracellular matrix. But in a tumor, just remember, it's a lot of cells. So therefore, and I will, I agree you can call it cell plus cell plus cell, but I just have to call it a tissue. That is what a tissue is. A tissue is a cells that are all joined together, held together. And so those are, that would be your tissue. If there was, in this scenario, in this tumor, if there was a lot of fluid, etc., then I don't know how that they would do that sectioning that I just showed you, in that cassette. Um, so everything that we talked about in the stroma yesterday, all that should be there. Can you identify the macrophage? That's another story. Can you identify the T cells are circulating in the blood? You cannot see them. So if you cannot identify, can you identify the fibroblast? Then that's another story. So so those are the ones that are here. But remember, this is like the core of the tissue. So it is. This is on the inside here, and it's not on the edge. So I hope I was able to answer your question. And so from here, let's. So yes. So this is like a digital kind of a created image, the ANC. But then when it was matched with histology, and staining, which is B and D, it seemed that they kind of like matched up. And how? So I want you to look at malignant, the ones that are malignant here, and you can see that in this kind of optical kind of technology here, that they had these, they showed up exactly like a X-ray, like the opposite. They showed up as white patches, and that white patch matches up with the other one here. So when they did histology, so these are nice experiments where they cut thin sections, and they took one section and put it here, and the other section, they were able to do histology, and so they kind of like match up. So that's what they're trying to show with these red arrows is a matchup. And so these are locations uh, of cell growth. These are locations of these tumor cells that are being surrounded surrounded by stuff. So here also look at the normal cells. In the normal cell, this is just like really hard to tell what's going on here. And so therefore they have put in arrows for the normal cells to be able to identify the locations. And in this optical kind of image, you see where I have put in this round circle, this is less dark. It's more whitish. So one more time, take a look. This is more whitish, and what they're saying is it is in this location that it is slightly darker. So this is the histology image. And so anything that is done by digital smart means has to be, at the end of the day, kind of it has to be verified by a normal histology stain. So uh, and so I believe they might have used also acridine orange there, which takes up acridine orange. The more the cells multiply, the more the more acridine orange is taken up. It goes and intercalates in the DNA. So so it goes and intercalates into the DNA, and so you get much more than just orange colored, dark color. And so let's go to the um the next one is a confocal microscopy image, and in this one um this one is it's a mosaic. A confocal microscopy, um you know a confocal microscopy, confocal microscopy in itself is a taking a slightly more a bigger section or slightly thicker section, and it can take layers and layers and layers of each section. So it takes the first layer, then it takes the image of the second layer, and then the third layer, and then the fourth layer. All right, and I'm not talking about a huge tumor and not a whole human body, but a slightly thicker piece or a slightly smaller piece, and then it will reconstruct. So in confocal microscopy, it will reconstruct every layer, and it will try to give you an image that's kind of like, it is not a 3D uh, image. It still looks like a slide, but it'll have reconstructed. So it's not going to give you a colorful 3D looking image, but it will give you a basic idea of it. And so, so this one is a basal cell carcinoma, and so this one is the uh, confocal mosaic image. Confocal mosaic image means all the layers have been joined together, and they have uh, given a somewhat 3D effect. There's always some 3D touch to it, but it's not a lot. And so they are able to show that there are different locations, and they've combined it with other things to be able to show that there is a different. This is, I think, in later on to show you that there is some kind of a region here. This is on top of the image, and they're just saying that they are finding that basal cell carcinoma cells um, can be detected, and the member is being shown in the H&E stain. And so they're saying that this is, they're finding that this is a darker region, and so that's the arrow that they are showing you, and that this is matching up with that light. So if you look at this section, this is much more lighter than the others, and they're saying that this is it. It's a little hard for me to also imagine that that's the case, but so be it. And so that correlates with the H&E. So this is your H&E. And again, remember in H&E, if you have too much of those Again, I would think that this is also a location, that this is also a location. And so this one is the frozen tissue section. What is called mm, again
Recording in
fluorescence,
progress.
you have to have UV on one side, and they're obtaining by this methodology called as muse. But then again, they have to always revalidate it by using H and E. Oh, sorry, H and E is here. And so they have to validate this with H and E. And so A is a um, this is a converted from the program. This is a converted image, and so this one is the converted image. Um, this is the virtual H&E image, and so all they're saying is that if you look at so this is all the cytoplasm area. These are all the cytoplasm, and these are the nucleuses. And they're in a line here, so I would not say that this is just a regular tissue. Everything looks fine and dandy here, and they're just trying to match it up. They're trying to match it up with different locations here. And so I'm just giving you this as an example of not a cancer, but using um, digital features and virtual imaging, conversion to virtual imaging that they verify with conventional histology. So essentially, what I'm trying to say is that people are trying to take the conventional one and then convert it into various means, and then they're trying to see if it matches up. So if I get this kind of a virtual image, then how am I going to match it up to this one? So that's pretty much it. So it gets more complicated here now. Um, Looks great, but very, very, very complicated. I think imaging is probably the most um, fascinating, fascinating, and it's endless learning. So now I'm talking about stereo projection. I think is my time over, Dr. Bhomik? No. Okay, I will just continue and try to finish quickly. So, where was I? Yes. So, stereo projection, again, confocal microscopy takes in layers, and but now this is even bigger. Okay, so this is a mouse GI tract, and they have still stained the cells with orange with propylium iodide. They have stained with orange, so anything that's orange or little orange yellow spots that are here are propylium iodide, and they have stained the um, which one is the gray here? So the remaining, propionimide is just the nucleuses, and the cells are all stained gray. So anyway, to make a long thing short, stereo projection is trying to just give it a full 3D look. And so here is a villi that I had shown you before the other day in a video. And so those are the crypts. Those are the crypts. Remember, the crypts are down here. The villi is up there. And so this is trying to give you the 3D along with confocal, that's trying to give you the 3D projection. And so I think according to this one, the sections that are, um, I don't know where I wrote that, but those sections the sections that are green, so here, this green, these are sections that are found to be kind of abnormal um, in this location. So these are all severe, very much digital. Again, they are all confirmed later on by sections individually. So illuminal scan and serosal scan uh, for for stereo projections. Explaining this is much more harder. Maybe we can in the future take some time just to go through the full methodology that is required to take this, to understand this, and the full methodology and the full three imaging system that makes it 3D, and that would be better. And the rest, as you know already, it's AI assisted pathology. The rest is all AI assisted, and so these are things that you are already learning from Dr. Bhomik. And so these are taking your H&E stains and trying to convert it, um, and trying to look at any remember PDL, we were talking about certain um, uh, s- uh, the certain markers that they might have. And so with H&E stain, people are taking that and then they're, trying to do some, a lot of machine learning through this. And so we're not just doing the H&E stain, but we're also using in these virtual IHCs, they're taking these molecular markers and not doing the H&E stain and just taking the molecular markers and then recreating a immunohistochemistry. And so it's called as a virtual immunohistochemistry. I have no idea how that would be, but that's what people are trying to do. So we're like taking things and going backward and trying to recreate. And so some of these methodologies are I just like to say that these are things there. So again, a lot of challenges are there. How do you explain it? How do you explain, understand the virtual stuff, and how do you integrate it? I just wanted to add some other names, just throw in these other names, and that is your high dimensional spatial omics, okay? New ones. And these are the things that, you are now combining, combining the stained ones with immunofluorescence, with antibodies, and you're looking at not only the cells, but you're looking at 10 to 40 proteins, and you're mapping them together. And then it is called as, I don't know why it's called as iterative staining. It's probably just looking looking at various staining tissues and then compiling it all together with the proteins. And so these are all spatial. So spatial omics, what it does is, I have not written it here, I took it away, is it is giving you a location as to where these cells stand within each other. So it gives you a microarchitecture of which immune cells are on the top, which immune cells are at the bottom, what are they doing based on also staining their proteins. Just like yesterday or the day before, I showed you that we have to stain the proteins to know whether that's the real cell. Mass cytometry also, again, you use, just like electron microscopy, you use metal tagged antibodies, and you get very, very, um, you can get much more details with the 40 different markers. So everything is spatial here. It can be combined with DNA. It can be combined with DNA and RNA and gene expression, and made into an image that combines everything. So it's kind of like whatever, I talked about the collagen assay. You convert the collagen assay into some kind of a image, and then you align it with an H&E layer. So at the end of the day, the H&E stain is the master. You still need to do that. Okay. So all of these have a lot of clinical potential. Okay. And cells, they kind of like go under evolution. These cancer cells, these tumor cells, they are also changing. So in one tumor, you will have different kinds of cells. The inside might be its original cell. The second, outside the tumor, is evolving, and it has certain properties. Maybe it will secrete something that breaks down the ECM, and then the third cell outside might have different properties. So these are called as all clonal evolution. And so if there's a possible to use all of these markers and to look for in the tumors, to look for the evolving tumor cells. All right, so that's just so sophisticated. Anyway, the bottom line is, what a simple, if I bring things back to simplicity, um, it's always about the interpretation as to whether it's um, what grade it is, how normal it normal, is it, and what grade of the tumor it could be. And if we come back to simple stuff, we are, you know, uh, all of these, the at the basic level, everything has to match up. So the bottom line is that things have to match up. The molecular markers, the immunohistochemistry, everything has to match up for it to make that interpretation. If you have too much dark cells, but they don't stain for Ki67, then that doesn't make sense. Ki67 is for proliferation. The the DNA has to show up. But if you find a lot of nuclei stain in the H&E, then that doesn't make up. So um, all of these. So I just kept these as additional pictures. I didn't have the time to do all of them, but just to show you that you check for borders, check for neoplastic tissue, and check for non-neoplastic tissue. And again, your methods are counting by area, counting by cell, counting by mucin production. Okay. And you know that colorectal cancer is one of my favorite ones. Remember, I showed you the crypts, etc. And so these are some of the crypts. These are pretty normal crypts here. These are fairly normal crypts. These are fairly normal crypts. Whereas it seems to be that these are getting more and more abnormal. So we talked about aberrant crypt foci. So these are getting more normal, while this is fairly normal tissue here. Whereas on the tumor side, let's look at it. I don't see any crypts, proper crypts. The staining is much more darker in many places. I don't see a pattern. I don't see the beautiful pattern like I saw here. I see a lot of more nucleus, So you barely see any cells. So you see a lot of small nucleuses, and very little cytoplasm. I hope I didn't say the wrong words. So you see much more
Recording in progress.
mucin, and so this one has hordes of mucin.